have yet to sign up for the chemistry picnic, it is this Thursday, May 19th, and Daniel May, and that's going to be at 5 p.m. And that's City Park. So, I believe the sign-up sheet is up right now, and it's going to come down after this talk or something like that, so we can get the right amount of food. So if you have yet to sign up, this is your chance. Also, Sam is just one of 17 people giving talks this term, and it's actually almost all in the next 2 weeks. And so that we have 2 tomorrow, starting at 3:10 with, or no. Oh, sorry, it's Jenna. Sorry, I didn't mean to panic you. So we have 2 tomorrow, 1 on Thursday, and then 2 on Friday, each starting at 3:10. So if you're interested in coming to more of them, we would enjoy having you. Check the university calendar for that 3 o'clock time slot, and you can find out who's when and where. It's been a pleasure having Sam in class multiple times. I'm kind of like her pseudo-advisor, pseudo-mentor, when her real advisor is like on sabbatical. So anyhow, hopefully I've given her proper advice. She hails from Potter, Wisconsin. It's been fun to have in class. Although she gets stressed out once in a while, but it turns out just fine, as she's discovered. She is going to be going to graduate school at the University of Madison, Wisconsin in chemistry. And without further ado, I will let you turn it over to Sam to talk about her research.
So like you said, my name is Samantha Knott. I'm a senior biochemistry major here at Lawrence University. And today I'll be presenting on transcriptional regulation of PrsA2 and Listeria monocytogenes. And that's a lot, but I'll break that down as I go through my talk. So hopefully you'll understand the title more when I'm done. And this is the work I did, or the research I did this past summer at the UIC College of Medicine, where I worked in the Freytag lab. And Nancy Freytag is a microbiologist, and she studies infectious diseases primarily, so bacteria that have the ability to cause infection and compromise the immune system. So to begin with, if you've heard of Listeria monocytogenes before, it's probably been in the news. So Listeria, in the simplest context, is a bacteria, and it has led to some of the biggest food recalls in the past century across the country and caused illness as a result. And right now, actually, Whole Foods just recalled a bunch of its chicken fried rice because it was contaminated with the bacteria I'm talking about today. And this happens because Listeria has the ability to grow in refrigerated food items, which is rather unique because most people put their food in the refrigerator because they don't want it to become contaminated with bacteria. And upon eating contaminated food, it causes listeriosis, which is a simple, I guess that's a complicated word for food poisoning. And this primarily affects immunocompromised individuals, meaning that most of us, if we were infected, would be able to fight it off and recover relatively quickly. However, if you're immunocompromised, which can mean you have an autoimmune disease, are pregnant, or are elderly, you are much more likely to suffer the consequences of it, which are bacterial meningitis and miscarriages in the case of pregnancy. And right now there's a whopping 30% lethality, which is quite high. And this research, one of the goals is to develop more therapeutics for a greater understanding of pathogenesis. In other words, we need to understand pathogenesis more, or the way it causes disease, so how it goes from outside of the body to inside of the body, making you ill, in order to develop drugs that will better combat infection and hopefully decrease this lethality rate. So Listeria monocytogenes looks like this. It's a rod-shaped bacteria, and it's, like I said, an opportunistic pathogen, meaning upon opportunity it will take over and compromise your immune system. It's also intracellular, meaning that it likes to grow inside of your cells rather than outside of your cells. And it's gram-positive, meaning that it has this thick layer of proteins around it that just protect it from its outside environment. And it also has an actin tail, which are these long, skinny projections, which are also called flagella sometimes, and these allow the bacteria to be motile, or move throughout its environment, either outside of your body or inside of your body from cell to cell. And Listeria is primarily found in soil and fecal matter, meaning that through simple agricultural processes, this is often how food is contaminated. And like I said, it lives in different environments and withstands stress. So it's unique in the fact that it can live in high salt concentrations, high sugar concentrations, outside of your body, inside of your body, at different temperatures. Almost anything you can think of, it's going to withstand that environment and continue to be able to replicate without dying or being degraded. So in order to understand the pathogenesis, we're going to look at Listeria life cycle. So don't worry about all these little names. They're not important right now. I'll get into that later in my talk. However, in general, Listeria is found outside of the body, and changes in environment trigger Listeria to go from its non-virulent to its virulent state, meaning it goes from outside of the body to inside of the body. And upon this, it induces a gene called PrsA2. And PrsA2 is crucial for it to go from the non-virulent, which doesn't make you sick, to the virulent strain that makes you sick. And this gets activated, and it makes it triggered to the virulent form because it makes all of these proteins, which are also called virulence factors. And these virulence factors are what make you feel sick. They're what destroy your cells, and they are what allow Listeria to continue to replicate without your immune system getting rid of the bacteria from the body. So in the simplest terms, it wants to get in, so it can go from that non-virulent to the virulent state. It wants to make these virulence factors or proteins, and then it wants to get out. So after it makes these proteins, it goes to the next cell because it wants to continue to use your resources. So it spreads from cell to cell to cell, and this is how an infection spreads. So like I said, virulence factors are critical for pathogenesis, or critical for making you ill. And they allow for creation of a niche in the host. So like I just said, these proteins help Listeria able to withstand the inside of our body and thrive. And they also are considered the toxins and enzymes that make you sick. So often people will throw up profusely when they have food poisoning, and that's because these bacteria release toxins that make you very ill and vomit. And then these virulence factors also damage the cells, like I said, and they hijack and invade the immune system. So they not only live within your cells and replicate, but they hide from our immune system so that we're not able to get rid of the infection immediately. Okay. And I'm going to go over how this is happening at a molecular level now. In other words, we know that there's this environmental activation, or from the non-virulent to the virulent state, that happens through activation of the gene PrsA2. And if you remember, I just said PrsA2 encodes the virulence factors, or the proteins, which are what make you ill. And if we start up here, we know, or maybe you know, Listeria has DNA like all other organisms, and this DNA is the blueprint for the cell. It contains all the information to make all the proteins and gene products and chemicals that the bacteria needs. And if it wants to make its virulence factors, it's going to have to be transcribed or made into RNA first. So this is the process of transcription, where you make a copy of the DNA and create an RNA. And this happens because this DNA is the blueprint to your cell. It's your really important document. You don't want to just create proteins from this DNA because it might get damaged and you don't have any more of it. You don't want to accidentally make a mistake and destroy your DNA. So you make this intermediate RNA, and then this later contains the message or all the information to make the protein. So later on, it'll make the proteins or the virulence factors. And if you go up here, I just want to point out that in order for it to make a copy of the DNA, it requires this RNA polymerase. An RNA polymerase is the machine that starts making the copy from the DNA. And the way it binds is through this sigma factor, And just remember that this is essential. Without this sigma factor, transcription won't happen, and you'll never have proteins made or virulence factors made. And PrsA, when this process is underwent, it's not expressed alone. There's another protein expressed called PrsA2, which is the focus of my talk today. And PrsA2 is a secretion chaperone, and it's a molecular chaperone for virulence factors, and it folds them, which is essentially taking these amino acids coded for from the genes and making them into functional proteins or virulence factors. And this has to happen because here you have these long skinny chains of amino acids. However, they are virtually non-functional in the body, unfolded, meaning that they are like the small children in our bodies. The cell is a really big, complicated, crowded place, and these amino acid chains don't know what to do. They don't know where to go, what their job is, or what place they're supposed to be in the cell. So in order for them to become functional proteins or virulence factors that make you sick, they need a molecular chaperone, which is PrsA2. And PrsA2, like I said, just folds the amino acid chain so that it can become a functional folded protein. And right here it's important to point out that unfolded proteins or virulence factors are not functional. In other ways, they aren't going to be able to make you ill. So like I said, PrsA2 is a secreted chaperone required for pathogenesis, or the ability to make you sick, but also cell wall integrity and a variety of other functions. So it folds the proteins, but it is also responsible for secretion of the proteins and something called cell wall integrity. So in other words, they make a bunch of proteins that help the Listeria or the bug withstand stress or withstand its environment. And part of this is the reason why lots of strains of Listeria are antibiotic resistant, because they have the ability to withstand penicillin, which normally breaks down their cell wall. And then PrsA2 also folds the proteins for flagella, so those long skinny projections on the cell that are responsible for it being motile or moving through the cell. So we know that if you take PrsA2 out, virulence factors are not functional, and that regulation of PrsA2 is not known, meaning that we want to look at how is it expressed and how we can possibly control how much of it is expressed. Because we know that depending on how much PrsA2 is expressed is going to determine how many virulence factors there are, because those long skinny chains of amino acids need to be folded. So it's a great target, because if we could potentially cause PrsA2 to not be transcribed at all, we would think that there would be no virulence factors, or Listeria would be unable to cause illness then. So the hypothesis is if you prevent PrsA2 from being transcribed, will functional virulence factors be made? In other words, if you prevent it from being expressed, will you have these long skinny chains of amino acids that are basically non-functional and useless for the cell? Or will you have these functional folded virulence factors or proteins that have the ability to make you sick? And the way that you can look at this and potentially regulating this is looking at that process I described before called transcription, or making the copy from the DNA that eventually goes on to make proteins or virulence factors. So this is the PrsA2 gene, and this is the DNA. And right here, this arrow is the start of transcription, or the start of where they make the copy that's going to go on to make virulence factors. And right here, a postdoc in my lab actually looked upstream of this gene. So if you look at right before a gene is transcribed, there are areas on the DNA that can potentially dictate or regulate how much of PrsA2 is expressed. So the goal of this study is to look at specifically these regions labeled A, P, B, and S, and assess their ability to alter PrsA2 expression and assess its ability to alter PrsA2 expression under different conditions. Because also gene expression can change upon your environment. So maybe these are useful in one environment for regulating the amount of PrsA2 made, or maybe they're not in a different environment. So if we go over this again, what I just showed are regulatory elements, which act as molecular switches. So they control the amount of PrsA2 that is being made, so they act to control the amount of virulence factors that are being made. And there's three possibilities. There's the possibility that you can take out one or two of these promoter region elements, and then you will have no functional virulence factors because PrsA2 is not being transcribed. Or there is a potential that you can take out maybe just one of them, and a little bit of functional PrsA2 is being made, so as a result a little bit of functional virulence factors are being made. And then another option is that maybe you don't take out any of them and you have some amount of functional virulence proteins or virulence factors that are being made. So the methods for this experiment was that I created mutant strains or versions of Listeria where I either took out one, two, or multiple of those upstream regions, and then I wanted to look at what happens to PrsA2 expression, because we're hoping that maybe there's a potential that PrsA2 won't be expressed at all in one of these strains, which means that they wouldn't create virulence factors and Listeria would not cause illness. And the way I looked at PrsA2 expression was through phenotypic assays. And that's a fancy word for saying I was looking at what does the bacteria do when I alter it? What is the personality of it, so to speak? How much protein is being made and how much PrsA2 is being made. And I can either measure the expression of PrsA2 directly, or like I said, I can look at the virulence factor production, because virulence factors will not be functional if PrsA2 is not being transcribed, because there's nothing to fold them. If PrsA2 is being transcribed, you'll have these functional proteins that make you sick because PrsA2 is there to fold them up into their functional state. And then lastly, I was looking at how is this affected by different environments, especially stressful ones. So like I said, this can alter in different environments. So maybe it's altered in a high salt concentration in your body or at a different temperature in your body. So I have three controls that I used for this experiment. And the first one will be wild-type PrsA2, or the normal allele. And this is a fancy way of saying that I had the Listeria with the PrsA2 gene, and I did not alter it at all. I didn't take out any of its upstream regulatory factors. And this is a standard for expression of PrsA2. So you would expect normal virulence factor production. So the normal amount of PrsA2 folding that's in Listeria in the environment. And then I had a strain where I eliminated PrsA2 completely. So that little triangle just means that I deleted the gene entirely. And you would expect here that there will be no virulence factor production because virulence factors won't be folded, PrsA2 isn't there. And then lastly, I had the sigma factor of PrsA2. So if you remember, that was the essential part of transcription. So it's essential for PrsA2 to be transcribed or to be made. And once again, you wouldn't expect any virulence factor production because PrsA2 won't be transcribed without the sigma factor. And then lastly, I had my mutants. So the triangle just means I deleted the A portion, or the AB portion, or all of the portions upstream of PrsA2, the gene. And this is a question mark. I don't know what was going to happen, and that's what I tested in this experiment. I wanted to look at how much PrsA2 is being made when I deleted these upstream regions. And there's two options. I could either have normal expression that's equivalent to wild type. So So this would mean there would still be functional virulence factors being made, or I could have abnormal, more or less expression to wild type. So here you see I can either have these non-functional useless amino acids when I take out these portions, or I might have functional virulence factors still being made. And this is my first set of results. So the first thing I did was I looked at the amount of PrsA2 being transcribed directly. In other words, I can take the DNA with the gene. The promoter is just the start of transcription, and this arrow indicates that. And I can fuse this gene to another gene called Gus. And Gus is just derived from another bacteria, and it causes fluorescence or glowing, meaning that when PrsA2 is expressed, Gus will be expressed. So I can measure how much of the chaperone that folds virulence factor is being transcribed or expressed by looking at how much fluorescence there is, because when PrsA2 is transcribed, Gus is transcribed. And right here, I have my upstream regions. So here I have listed my mutants. And once again, that's just where I either got rid of one, two, or multiple of these upstream sites. And the axes of this, right here, I have Gus activity. So that just means how much did it fluoresce. So you go from 0 fluorescence, which would indicate no PrsA2 expression, or you go from 40 units, which would indicate a lot of fluorescence or PrsA2 expression. And down here, I have incubation. So I took measurements at 2, 4, and 6 hours because environment can change expression, but time also can, meaning that genes or bacteria can cause genes to be expressed higher or lower at certain time points. And if you see here, there's a general trend here where there's a lot of PrsA2 made at the 2-hour incubation time, and then it kind of goes down at 4 hours, and it goes back up at 6 hours. And this red one is my wild type right here. So if you compare the other strains, you see that PrsA2 expression is relatively the same at the 2, 4, and 6 hour time point. And this indicates that LM or Listeria mutants maintain PrsA2 expression equivalent to wild type, meaning that I can delete multiple of these upstream promoter regions and still have fully functioning virulence factors as wild type over here. Meaning that you can delete these upstream factors and it will be just as virulent. It will still be folding just as many virulence factors because PrsA2 is being transcribed equivalent to wild type. And this was shown in several sets of mutants. So I showed just a few, but I tested this with 30 different combinations of upstream regions. So quite a bit, and they all showed the exact same trend. And the next thing I looked at was the folding of a virulence factor. So PrsA2 expression can be measured by looking at the amount of virulence factors as well, because there won't be any if PrsA2 isn't there to fold them. And I looked at phospholipase in particular, and phospholipase hydrolyzes fat and fatty acids, which pretty much just means it breaks down membranes. And this is important when you look at the process of phagocytosis. So like I said, our immune system does try to eliminate the bacteria from our body, and it does that through phagocytosis, which is essentially where you just have these immune cells or police, and they will go around the body and catch the intruders or the bacteria. So they'll eat the Listeria, literally, and send it off to degradation. And phospholipase is excreted by Listeria so that it can beat up the cops and escape the police and go on and run rampant through the body, so it can continue to replicate cells and use its resources. In other words, it evades our immune cells and it evades our body's way to eliminate infection. So when I looked at phospholipase activity, I once again looked at my wild type, and what I did was I struck my bacteria on plates with charcoal, and you can tell how much phospholipase was excreted by the bacteria by looking at the area of clearance around it. So the plate mimics our cell membranes because it is rich in fatty acids. So if phospholipase is excreted, it will cause this area of clearance around where the bacteria was struck, meaning it was excreting this enzyme that broke down the agar plate. In wild type, you see here that we have this white area of clearance, or a baseline for how much phospholipase is normally there in Listeria. And you can see here in our PrsA2 where you delete the gene completely, and the -10 PrsA2 where I delete that sigma factor, we don't see any phospholipase activity, which makes sense because PrsA2 would not be transcribed, which means there was nothing to fold this virulence factor that breaks down membranes. And then down here, I have my mutants where I, once again, either deleted 1, 2, or multiple of those upstream regions, and then I assessed activity. And here you see again, in all 3 of these, that the area of clearance around the bacteria is pretty much identical to the wild type strain, which indicates that, once again, you can delete 1, 2, or multiple of these upstream promoter regions, and there's still equivalent PrsA2 expression, or there's still just as much phospholipase activity, which means the Listeria is still able to make you just as sick as the wild type or unaltered version. So to go over this, they maintain expression equivalent to wild type, and it's dependent on multiple upstream regions, meaning there's this redundancy, which we also saw in the previous test. And this indicates that the bacteria really wants its virulence factors to be made. It has a bunch of regions upstream to make sure that they are made and to make sure that it stays in its virulent state. And once again, this was shown in several various sets of mutants. So not just the one tested, but approximately 30 different combinations of these upstream regions. So I looked at one other virulence factor called ACT-A, and ACT-A synthesizes the actin tail that you saw before, or the flagella that allows the bacteria to be mobile or move throughout the cell. And this allows for cell-to-cell spread, so it allows the bacteria to be in one cell and spread to another cell and another and another, and this is how an infection spreads across your body. And there are advantages to this because they can be allowed to continue to replicate in your cells and use your resources. It doesn't just want to stay put in one cell. So the way I assessed how much motility there was, or how much ACT-A there was by looking at how far the bacteria was able to move on a plate. So you can stick the bacteria in an agar plate and look at how big the colony is, or how big the diameter of the bacteria is. And that indicates how far it has moved out or moved through the agar plate, which would be equivalent to how much PrsA2 there was to fold it. So here we see my wild type strain, where I have a diameter at approximately 23 millimeters, which meaning this is how much Listeria is normally able to move or be mobile throughout the body. I then have those negative controls, or where I deleted the gene completely so it shouldn't be described, or I deleted the sigma factor so PrsA2 will also not be transcribed. And you see, similar to before, that there's much less activity than wild type, but there is still some activity. However, the colonies are very abnormal, and they're clearly much smaller than when you have the gene there. And then lastly, you have the deletions, where I deleted 1, 2, or multiple upstream sites. And here I see colonies that are almost essentially equivalent. So there is no statistical difference between any of these strains, which once again indicates that expression was equivalent to wild type. I could delete 1, 2, or multiple of these upstream regions and still have just as much motility as the wild type, or the normal strain of Listeria. And this was once again shown in several different mutants. So the summary is that PrsA2 expression is dependent on multiple upstream promoter area regions. So there's redundancy. And this makes sense because Listeria really wants to make sure that it makes these virulence factors that make you ill. Unfortunately, this means that PrsA2 expression is more complicated than we thought. And this was seen in the alcohol response. So we tested this in different conditions, which I didn't show today. However, we saw essentially the same result in 10 different conditions. The one condition that was different was the alcohol response, which I'll show you right now. So we grew the bacteria in 8% methanol, and most bacteria, this is a very stressful environment. We use alcohols as disinfectants, so they'll normally kill the bacteria. However, we see in all of these that these are not the same like we saw in the other virulence factor assays, that you can delete one, two, or multiple, and there is a quite drastic difference in activity or growth. So here we're measuring growth, which I forgot to mention. And you can also see that the error bars are very big, meaning that maybe this promoter region regulatory region is responsive to alcohol. So maybe alcohol is what causes one of these promoter regions to be important. So the conclusion is that Listeria maintained expression of virulence factors. It had just as much of an ability to make you sick, even when I tried deleting those upstream regions to see what would happen. And LM, or Listeria, appears to have a diverse range of transcriptional regulation, meaning it has lots of different ways to make sure that it stays in its virulent state and makes these proteins that allow it to spread and grow and thrive. So future work, this was one of the first studies that looked at actually regulating this activity of folding of virulence factors rather than regulating just its expression. And this requires a starting point for small molecule inhibitors. So like I said, if we had an upstream region where I eliminated it and we didn't see PrsA2 expression, that would be really good, because that would mean there would be nothing to fold virulence factors, and you could potentially inhibit virulence factors from being made in the first place. And then also, future work will look at additional upstream promoter region areas. So you may have saw that I eliminated the gene completely, and there was still some activity of the virulence factors, which isn't expected. If the gene's not there, how was any folded at all? And this may be explained by looking at upstream elements. Maybe there is another gene that encodes a chaperone that folds virulence factors. Or maybe there's other genes that control PrsA2's expression that we just don't know of yet. So I'd like to acknowledge Dr. Hall for helping me put together this lovely presentation, and also Dr. Freitag, Dr. Laddie Cahoon, and Bobby Zatharath. They were the postdocs I worked with in the lab, and the USC College of Medicine, which via the Great Lakes grant and the McNair Scholars Program, I was able to fund my research this past summer.
Also, have you or your peers in your lab looked at to infect this bacteria in model organisms at all?
Yeah, so actually there's additional data to this. I didn't do it, my postdoc did it, but we work with mice. So you can look at viral counts or bacterial counts after you eliminate the gene. So there's also a virulence factor that causes Listeria to hyperactivate its growth, so be able to grow at hyper high rates, I guess, very high rates. And you see that if you eliminate PrsA2, growth is lower. However, there's still a lot of growth compared to the non-virulent strain.
How do you know that the upstream regions are deleted?
How did I know? I didn't actually construct the regions. I didn't actually construct the mutants. However, I believe they were sequenced after the deletions.
And the plot that you had where you were looking at the fluorescence from the gust thing to make sure PrsA2 was there, you did that for 2, 4, and 6 hours. I'm curious what influenced you to choose that time scale, and if you go into farther time, would you see, or what would you anticipate seeing, like if you let it sit for longer and incubate for longer?
Yeah, so the experiments actually were initially preliminarily carried out up to, I think, 14 hours. And at that point, you just see it kind of level off. It doesn't really change at all then, it just levels off.
So there is something then to that weird 4 hour, because since your data showed that they all went down a little bit in expression at that 4 hour mark, I give up what's going on there, because then they go back up.
Yeah, well, I think it might be just more to the sensitivity of the assay. So at 2 hours, that time point isn't completely accurate because there's not very many bacteria there. So sometimes it counts more expression than there actually is, and that it's probably actually closer to below that 4 hour time point.
How did you not get Listeria? I mean, you just saw how horrible it was, and you were working with wild type strains. Why did you throw up a lot during the research?
No, like I said, you'd have to be autoimmune compromised for it to really affect you. And I didn't eat the bacteria that was found with them, so I didn't have an option. One of my postdocs was actually pregnant, and her doctor thought she was crazy, so I don't know why she kept doing it.
She didn't get sick either.
No. I mean, they're attenuated in a way that the research still works, but they're not going to be able to be as hypervirulent as they normally are.
And in all the data that you saw while you were doing your summer research, is there any data that you saw that you were surprised by, or kind of like shifted the direction of the project?
I guess I was surprised by the alcohol response I saw, just because it was different. I mean, all my other results were virtually exactly the same. It's still very virulent even when I tried to make it less virulent, and the alcohol was very abnormal. The error bars were all over the place, so some days I would try growing Listeria in the alcohol and it wouldn't grow at all, and then other days it would grow better than wild type when you deleted upstream promoter regions. So I think there's just something upstream that we have no idea of yet that's causing that. And some bacteria are also alcohol responsive, which is really interesting. And attributes to, I think Listeria now, they're having issues disinfecting it in factories even with alcohol. It's becoming a little bit resistant.
Not entirely sure if this is relevant considering your findings, but did you have trouble with autofluorescence when you were doing the GUS assays?
Yeah, like I was saying, it's very sensitive, and I was using kind of an old fluorimeter, which I'm not sure if matters, but I just didn't know if it was entirely accurate at low bacterial counts. So I guess that's my answer. It's a very sensitive assay. So the results I showed, I actually ran all of the strains 3 times each trial, and I ran 4 trials. So there's a lot of data points there, and the error bars are still very high.
So what I mean is, since you didn't actually make the mutants, you might not have been able to, but were there mutants available that didn't have the GUS, but everything else was the same, so you could compare the autofluorescence?
Yeah, we had just a control.
Okay, well, thank you very much.